Hello, everyone, and welcome to today's webinar, How to Improve Patient Management with Molecular Solutions for Cervical Cancer Screening and CIN23 Diagnosed Women. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by QIAGEN. To learn more about our sponsor, visit QIAGEN.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them in the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would like to welcome our speakers, Anja Oštrbenk Valencak, Analyst in Laboratory Biomedicine, Institute of Microbiology and Immunology, Faculty of Medicine, University of Ljubljana, Slovenia. And Wieke Kremer, Resident Obstetrics and Gynaecology, Amsterdam University Medical Center. Anja, Wieke, welcome. You may now begin your presentation.
So hi, thank you for the nice introduction. As mentioned, my name is Anja, and I'm gonna show you today the clinical validation of the NeuMoDx HPV assays in cervical cancer screening. So this is my conflict of interest statement. I would like to start with 2030 goals set by WHO. We have two key prevention strategies available for elimination of cervical cancer. One is the vaccination against human papillomaviruses, and the other is very effective screening programs. That is, of course, followed by treatment of detected precancerous and cancerous lesions. So one of the goals is that 70% of women will be screened using a high-performance test twice in a lifetime, preferably by 35 and 45 years of age. So the next question that we need to ask is, what is a high-performance test? Our research group that is led by Mario Poljak has done inventories of commercially available HPV assays worldwide. The first one was performed in 2010, followed by 2012, 15, and 20. And as we can see, the number of HPV assays is increasing dramatically. And if we would be in 2010, we would only have to choose between 70 HPV assays. Unfortunately, in 2020, we have identified more than 150 HPV assays and more than 425 test variants. So the question which HPV assay to use is not as simple as we would like to be. So the main thing that we have discovered in our inventory is that approximately 60% of HPV assays on the global market are without a single peer-reviewed publication, and moreover, 80% of them are without published any analytical or clinical evaluation. And even more concerning is that more than 90% of HPV tests performance evaluations were not in line with standards that are agreed in HPV scientific community. So of the 600 HPV assays that are currently on the market, majority of them are not suitable for safe use in clinical settings. So from 2009, we have requirements of HPV assays that needs to be fulfilled so they can be used in primary cervical cancer screening. And the main three requirements are the clinical sensitivity for CIN2+, that is not less than 90% compared to the hybrid capture 2 in women that are older than 30 years, a clinical specificity for CIN2+, that is not less than 98% of clinical specificity of hybrid capture 2, and also that the assay performance is robust. So the reproducibility and interlaboratory agreement is met when compared to the standard assay. So the study that I'm going to present today, it started all in December 2009. So we have performed a national HPV prevalence study. For those of you who do not exactly know where Slovenia is, we are basically sitting in the heart of Europe. We are a very small European country. We have approximately 2 million people that are living here, and we actually have very effective nationally organized cervical cancer screening program with the coverage rates above 70%. And we have also implemented the HPV vaccination into a national immunization program, and we offer free of charge HPV vaccination with nonavalent vaccine to the girls in the 6th grade. Unfortunately, our coverage is not very good. It's about 50%, but we are hoping that this is going to improve in the following years. So the study that I mentioned was started in 2009, and we have invited women aged 20 to 64 years old who attended their routine screening visit. So we have a total of 4,400 women, and they are representative for Slovenian population-based cohort. We have obtained two cervical scrapes. One was for conventional cytology, that is standard of care in Slovenia, and the other one was cervical sample that was collected in ThinPrep medium. All the samples were then aliquoted and stored at -80 for further analysis. So the colposcopy and biopsy were done based on standard of care for Slovenia guidelines. So they were performed if cytology was ASCOS high or worse. But irrespective of that, we have encouraged gynecologists to perform colposcopy also if women were HPV-16 or HPV-18 positive. So for this particular study, we have randomly selected around 900 women that were above 30 years old. And we have tested them additionally with NeoMoDx HPV assay. So the original cohort was actually tested with 4 different clinically validated HPV assays, including hybrid capture 2, COBAS, and new Abbott's test, Alinity HPV. So maybe a few words about the NeoMoDx HPV assay. It's fully automated and high throughput random access platform. It integrates nucleic acid extraction, followed by amplification, detection, and automated reporting of results. It was launched 2 years ago and it is designed to detect 15 high-risk HPV genotypes. It can distinguish HPV 16 and 18 and report other 13 HPV genotypes as full signal. It also have internal process control that targets beta globin just to make sure that sample is adequate. So from our cohort of 900 women, we have divided samples on which we have assessed separately clinical specificity. This cohort were women that had 2 consecutive normal cytology results. So that means that women were included with baseline normal cytology, and then 3 years later, the cytology was normal again. The clinical sensitivity was assessed on 68 women that had underlying CIN2+ lesions. Out of 900 women, we have excluded 27 because of the invalid internal control, which means that in the final analysis, we have included 101 women in the clinical specificity and 67 women for clinical specificity. So first, I would like to present the comparison between NeuMoDx HPV assay and hybrid capture 2. As you all know, this is the standard comparator that is also incorporated in the Meijer guidelines. So as mentioned, 67 women with CIN2+ were included in the analysis. And as we can see, the relative sensitivity when compared NeuMoDx HPV to hybrid capture 2 is 0.98, and the non-inferiority testing is below the set. So the p-value is below 0.05, which is set for statistical significance. So with this, we can check the first Meijer requirement for HPV assay. The clinical specificity was assessed on 101 women. We have also additionally genotyped all the discordant result, but as you can see, very nice agreement was between hybrid capture 2 with a little bit more of hybrid capture 2 positive result and NeuMoDx HPV negative result, but that was kind of anticipated since hybrid capture is prone to the cross-reactivity with other genotypes. Overall, NeuMoDx was slightly more specific with relative specificity of 1.03, and also the p-value was below 0.05. With this, we have also checked the second requirement, and the last requirement was done as intralaboratory reproducibility. As you can see, extremely high kappa value was determined, 0.9, and overall percentage agreement is 95.7. Very similar results were obtained in the interlaboratory agreement, where we have sent samples to the Amsterdam, and we have obtained a kappa value of 0.86, and overall percentage agreement was almost 95%. So with these results, we have confirmed all the requirements for the new HPV assay, and we can, based on our results, we can say that NeuMoDx can be considered clinically validated, but we have set a bar a little bit higher as well. So what we have done is that we have compared the performance of NeuMoDx with other PCR-based clinically validated assays, starting with COBAS. But as you can see, very, very similar results. So practically sensitivity is the same as the one of COBAS. And we have set here COBAS as the standard comparator, but the non-inferiority criteria was met again. And also for the specificity in women above 30 years old, we have very low percent of discordant results, and the specificity of pneumodeix, the relative specificity was above 1, again indicating a bit higher specificity. We have repeated this analysis and compare it to the Alinity HPV assay. Again, the sensitivity of the pneumodeix was 0.97, and the non-inferiority criteria was met again. When the specificity was evaluated, we had similar percentage of discordant result as compared to COBAS testing. And the relative specificity was again slightly higher, 1.03, and also the non-inferiority criteria was met if Alinity would be considered as a standard comparator. I just plotted the results that I presented on this graph, just to briefly go through. So on the left side, we have sensitivity, and on the x-axis, we have 1 minus specificity. And these lines are 95% confidence interval lines, and all the assays that were compared in our study are very similar in performance. So to conclude, what I would like to send like a take-home message is that clinical sensitivity of pneumodeix for CN2 plus was comparable to that of hybrid capture to COBAS and Alinity, but the specificity was slightly higher compared to all other assays. The clinical sensitivity and specificity for CN2 were non-inferior to those of hybrid. The clinical sensitivity and specificity were as well non-inferior to COBAS and Alinity, and according for the intralaboratory and interlaboratory reproducibility, we have showed a very high overall percent agreement. So with this, we can say that pneumodeix HPV assay fulfills the international consensus guideline requirements and can be considered clinically validated and safe to use in primary cervical cancer screening. Maybe just a thought to conclude. So we have all been affected by COVID-19 pandemic, but maybe we should seek opportunities to not only that we have survived this, but we can use that to have more efficient prevention, and maybe by promoting more cost-effective protocols. And if we focus on women that are at high risk, and maybe discourage inefficient policies that are in some countries in the world, we could use the platforms that were established between the COVID-19 pandemic to use them to achieve the WHO 2030 goals for the elimination of cervical cancer. Yes, with this I will conclude. I need to acknowledge all the people that participated in the last 15 years in this study. It's been a huge work, and I am very grateful to all of them that we can have reliable data and do this analysis. And of course, if you have any questions, please let me know.
So, thank you all for listening. I will tell something about conservative management of SIN2 and SIN3 lesions based on methylation test results. These are my conflicts of interest. So, challenges in management of premalignant cervical lesions are mostly related to their heterogeneity. Cervical screening programs aim to detect and treat premalignant lesions, but not all of these would eventually progress to cervical cancer. Management of SIN is based on the histopathological grading, which mostly consists of surgical excision of CIN2+ lesions. This pathological grading is only moderately reproducible, of about 70%. Moreover, pathologists cannot distinguish lesions likely to regress from those likely to progress, causing overtreatment of regressive lesions. Particularly in young women, high regression rates have been described. About 20 to 50% of SIN2 and SIN3 lesions regress spontaneously, and in young women, up to 60% of SIN2 can regress. This is problematic because surgical treatment of SIN is associated with preterm birth due to cervical insufficiency. Therefore, there is a need for markers that distinguish lesions with a high progression risk to cervical cancer from those with a low progression risk. DNA methylation markers have this potential. Methylation of promoter regions of genes leads to gene inactivation. And when this occurs in promoter regions of tumor suppressor genes, this can lead to the loss of their tumor suppressive function. Promoter methylation of several genes is known to be involved in cervical carcinogenesis, and methylation levels of these genes increase with disease progression. This figure shows a schematic representation of cervical cancer development and the heterogeneity within premalignant lesions. Productive HPV infections can give rise to productive SIN lesions, which are mostly classified as SIN1 or SIN2. The majority of these lesions regress spontaneously when the HPV infection is cleared. With HPV persistence, the infection can become transforming and cause transforming high-grade cervical lesions, which are mostly classified as SIN2 or SIN3. Some of these can still regress spontaneously. Within these SIN2 and SIN3 lesions, we can distinguish early and advanced transforming lesions. Early transforming SIN2 and SIN3 lesions are defined by a less than 5-year persisting HPV infection and share characteristics with productive SIN1 and SIN2 lesions. Their methylation levels are low to moderately high, and they have only few copy number alterations. They often have HPV-E4 expression and no or very low staining of P16 and Ki67. Advanced transforming SIN2 and SIN3 lesions are defined by a more than 5-year persistent HPV infection and share characteristics with cervical cancer. They have a cancer-like methylation profile with very high methylation levels. They have many copy number alterations, no HPV-E4 expression, and high P16 Ki67 levels. We distinguish these productive low-grade SIN1 lesions, and about 50% of the SIN2 lesions are like these productive SIN1 lesions. They have low methylation. DNA methylation increases with lesion severity and is extremely high in cervical cancer. And that's why we think they can be of interest for management of SIN2 and SIN3. Over the years, several methylation assays have been discovered and assays have been developed, and they have been evaluated in cervical cancer and pre-malignant lesions. These are some commercial assays that exist, and they are developed for triage tests of high-risk HPV-positive women in cervical screening. Most research has been done with the QIAsure methylation test, which tests for methylation of FAM19A4 and MIR124-2. It's a PCR multiplex, and it's validated for both self-samples and cytology samples. The QIAsure methylation test measures methylation of these two genes and is a CE-IVD registered kit. Like I said, it's a quantitative multiplex methylation-specific PCR. It's applicable on self-samples and also cervical scrapes, and it has an automated data analysis and objective readout. The assay has been developed for triage of high-risk HPV-positive women in cervical screening and has been evaluated in various screening settings. In 2 large trials conducted within the Dutch screening program, it showed good clinical performance for the detection of SIN3+, which is comparable to that of cytology, as shown here in the table. It has also been validated in a European screening setting within the Valid Screen project, using samples of over 2,000 high-risk HPV-positive women aged 30 years and above. The sensitivity for SIN3 was 77% and sensitivity for cervical cancer was 95%. Approximately 50% of SIN2 lesions were detected in this study. The test has also shown very good sensitivity for cervical cancer. In a large worldwide series of cervical carcinomas, 98% tested methylation positive. This slide shows the performance of the methylation assay in high-risk HPV-positive self-samples. These are from a Dutch trial, the PROTECT trial, which tested 633 HPV-positive self-samples. And as you can see, it showed good performance for SIN3+ detection with sensitivity of 70% and specificity of 68%. And also in this study, all cervical carcinomas were detected. This shows the long-term safety for methylation analysis compared with cytology. This was 15-year follow-up data from the Babascam trial, showing the cumulative cancer incidence. HPV-positive women with a negative methylation test at baseline, represented by the black line, had a lower long-term cervical cancer risk compared with cytology-negative women, represented in the green line. We also evaluated the long-term safety for SIN3 and showed that a negative methylation test provides a similar long-term SIN3+ risk in HPV-positive women compared with a negative cytology test. More recent data show that methylation can also predict clinical outcome. Luvanto and colleagues showed that a positive methylation result with the S5 classifier predicts progression of SIN2. Hampel and colleagues, they evaluated FAM-MIR methylation in a retrospective series of samples from pregnant women with cervical cancer, progressive SIN3, regressive SIN3, and no SIN. And in this study, the likelihood of cervical cancer or progressive SIN3 in women with a negative methylation test was 0. And what I'll tell to you about in more detail is the CONSERVE study. And in this study, we showed that untreated SIN2 and SIN3 lesions with a negative methylation test have a very high spontaneous regression rate. So here you see the methods of the CONSERVE study. It was a study conducted in the Netherlands and included 114 women diagnosed with SIN2 and SIN3 on a cervical biopsy. These lesions had to be smaller than 50% of the cervix, and women were managed conservatively with 6 monthly follow-up visits for a period of 2 years. At baseline and during each follow-up, a self-sample, a cytology sample were collected and colposcopy was performed. Women who showed clinical progression during the study received surgical treatment. Progression was defined as exceeding the volume of more than 50%, a SIN2 that became a SIN3, or a SIN3 that became cancer. And also when the transformation zone was not visible, women had a treatment indication. Detection of adenocarcinoma in situ was also a reason to stop the study and have a treatment. Study endpoint was absence of SIN2 plus on histology or HPV negative normal cytology sample. This slide shows the baseline characteristics of the study. As you can see, we included mostly SIN2 lesions. 70% of women had a SIN2 and 30% had SIN3. Women were included mostly after 30 years old because this is the screening age in the Netherlands. But we also had 31 women who were under 30 years old. They were referred because of intermenstrual bleeding, for example. We mostly included smaller lesions, so 70% of all lesions were smaller than 25% of the cervix. About 50% of women were initially referred with high-grade cell cytology. The baseline methylation test was positive in 46% and negative in 52%. And here you can also see the HPV genotyping results showing that 50% of the women tested positive for HPV 16. So this slide shows the Kaplan-Meier analysis for endpoint regression. And as you can see is that methylation negative women showed more regression compared with methylation positive women, 75% compared to 51%. We found similar results in smears and in self-samples, and regression rates were not associated with SIN grades. This slide shows the combination of cytology with methylation and cytology with HPV 16 genotyping. Cytology by itself was not associated with clinical regression, but combined testing with cytology and methylation or HPV 16 was. So in the left figure, you see the combined effect of cytology with methylation. And as you can appreciate, in women with ASCAS or low cell cytology, methylation was significantly associated with regression. 88% of women with ASCAS low cell and a negative methylation test, this is the top blue line, showed clinical regression. In women with high-grade cytology, these are the dashed lines, methylation was not associated with regression. For HPV 16, we found the opposite. HPV 16 was nearly significantly associated with regression in women with high-grade cytological abnormalities, but not in women with ASCAS or low cell. We also evaluated the combination of methylation with HPV 16 genotyping. We found highest regression rate for HPV 16 negative and methylation negative lesions. This is the top dashed blue line. 85% of those women regressed. HPV 16 positive and methylation positive, the red line, showed lowest regression. Intermediate regression rates were found for women who tested either positive for methylation or HPV 16. This shows that there is a complementary effect of HPV 16 genotyping and methylation. So with the conserved study, we showed that you can use the Kaiser methylation test in management of women with SIN2 and SIN3. Women who are diagnosed with SIN2 and SIN3 and who wish to preserve their fertility, they could be tested for Kaiser, and if they test negative, this can reassure the clinician in active surveillance. Kaiser positive women could be advised to have excisional treatments. Another application for the QIAsure methylation test is a triage marker of HPV positive women with low-grade cytology, or ASCAS. With the introduction of HPV screening in the Netherlands, we saw an increase in colposcopy referrals, particularly of women with ASCAS and low-SIL. Triage of these women with methylation assay could reduce this over-referral, and only women with a positive methylation assay should be referred for colposcopy. Negative women could be rescreened after 1 year. You could also use a combination of ASCAS low-SIL triage of QIAsure with HPV 16/18, in which case, if women are double negative for QIAsure and HPV 16/18, the advice would be to rescreen within 3 years. A positive QIAsure test would be direct referral for colposcopy, and HPV 16/18 positive but QIAsure negative, the advice would be rescreen after 1 year. This application is supported by recent data from a post-hoc analysis within the Proboscam and FUSA-screen trials, which showed that the use of methylation can reduce the number of colposcopies. Triage of high-risk HPV-positive women with low-grade cytology using methylation could reduce the colposcopy referrals by 60%, while maintaining a sensitivity for SIN3 of about 70%. Using both partial genotyping and methylation led to a further reduction in colposcopy referrals, depending on the extent of genotyping, using only 16/18 or 16/18, 31, 33, and 45, up to 25% reduction in referrals. These figures show the SIN3+ risk following a negative or positive triage test with either methylation or 16/18 genotyping, or the combination on the right. The first figure illustrates that methylation negative women have an acceptable SIN3+ risk, supporting rescreening after 1 year. Methylation negative women have a SIN3+ risk of about 10%. HPV 16/18 negative women have a slightly higher SIN3+ risk of about 13%. And the lowest SIN3+ risk is among women with a negative methylation and a negative HPV genotyping test. 6.5% of those have SIN3+. So the last application I already showed in the beginning of my talk would be triage of high-risk HPV-positive women in cervical screening, which could be performed on self-samples or on cervical scrapes, and all HPV-positive women could be tested with QIAsure. If they're positive, they should be referred for colposcopy, and if they're negative, they should be rescreened within 3 years and referred for colposcopy if they are persistingly positive for HPV. Also here, there is an option of combining that with 16/18 genotyping. So to conclude, I've shown you that the QIAsure methylation test can be used for management of women with SIN2 and SIN3 and can prevent overtreatment and harm. When negative, this supports a conservative therapy, which is especially important in young women. Can also be used for triage of HPV-positive women with ASCAS or low-SIL cytology and prevents over-referral and over-treatment. When positive, it should instigate colposcopy referral. When negative, it gives a high reassurance against cervical cancer. And lastly, it can be used as triage marker for HPV-positive women in cervical screening, and it is an alternative to cytology, which is objective, reproducible, and can be performed on a self-sample. With this, I'd like to thank you for your attention, and I'd be happy to take any questions.
Thank you, Anja and Wieke, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, we have quite a few questions coming in already, so let's start here. Wieke, this question is for you. Do you think the assay is ready for clinical use? And how do you think it should be used in clinical practice?
Thank you, Christy, and thank you for this question. I think the assay is ready for clinical use by gynecologists, for example, who are doing colposcopy, especially as we see quite a few young women who are referred with HPV positive and minor abnormalities on cervical biopsies. They're often followed up for a long period of time, and often they receive treatment which can cause preterm birth in subsequent pregnancies. So I think especially in younger women, like under the age of 40, the methylation can really help to direct clinical management in younger women. So if the methylation is negative, it can support a wait-and-see policy in which you could say someone who's diagnosed with a SIN2 or a SIN3, you do follow up after 6 months up to 1 year to do colposcopy again to see whether the lesion is going to regress or not.
Thank you, Wieke. Anja, let's go with this question for you. Was assay evaluated on SurePath samples as well?
Yes, I haven't showed this data because the analysis was done elsewhere, but yeah, it was published actually together with our data. I think that approximately 1,000 women were included in that study. The pneumodic HPV assay was compared to the HPV risk assay from CellScreen. And the non-inferiority testing showed that the clinical sensitivity and clinical specificity met the threshold that they need to. So yes, we can. I can confirm that the assay is also validated on this type of samples. So it cannot be used. I mean, it can be used with either of the medium.
Thank you, Anja. Wieke, let's come back to you for this next question. Did you do separate analysis for SIN2 and SIN3?
Yes, thank you. So in the CONSERVE trial, we had 114 women, of whom 34 had a SIN3. So we had relatively small numbers of SIN3 for separate analysis, but we did do that separately. And we saw that in both SIN2 and in SIN3, methylation negative women have a higher regression rate compared to the methylation positive. So both in SIN2 and SIN3, we found a significant association.
Thank you, Wieke. And Anja, it looks like we have one more question, and we'll take this question for you. Is partial HPV 16/18 genotyping important in real-life clinical practice?
Yeah, thank you for the question. This could be a title for a new webinar, but to make it short, yeah, I feel that genotyping is a very nice way for the triage, but I think that we are not quite there to determine which genotypes should be separately identified. I'm a firm believer that the genotyping information is valuable only in the case that it affects clinical practice. So if the genotyping results is based on the risk that each genotype carries with it, I think that yes, then definitely we should use genotyping information, but I think it should be risk-based. So yeah, it's in short, yes, I think it's important, but in long version, it's a completely new session.
Thank you, Anja. And thank you, Wieke, too, for your time today and your important research. Do either of you have any final comments for our audience today?
Just thank you for being here.
Yeah, I also don't have any further comments, so thanks for listening.
Absolutely. Well, thank you for joining us, and we appreciate all the time and efforts that you're putting into this research. We also want to thank LabRoots, and we would like to thank our sponsor, QIAGEN, for underwriting today's educational webcast. Before we go, I also would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and additionally, those questions that are submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us. Until next time, take care and goodbye.